For this presentation, I chose high-content microscopy. I am Caitlin Murphy. High-content microscopy is a technique that combines automated microscopy with image analysis to obtain quantitative and detailed information about cellular structures and processes. It involves the use of fluorescent probes to label specific cellular components, allowing for visualization and analysis of multiple parameters simultaneously. In this method, cells are typically grown on glass slides or multi-well plates and treated with fluorescent dyes or antibodies to label specific targets, such as DNA or proteins, and are then imaged using a high-resolution microscope equipped with automated stage movement and image acquisition. So the acquired images are then analyzed using specialized software that can ID and quantify various cellular features like cell morphology, protein localization, and intensity measurements. So for my first image, it is showing control proteins affecting neurite outgrowth when overexpressed in PC12 cells, which were then transfected with GFP tagged ORFs. After incubation, they were stimulated with NGF for 24 hours and fixed and stained with Conne Alexa 647 to highlight the outline of the cells. So in the images, for A, it shows the phenotype of GFP. B, it is the control protein BAMP2. C, it's SNAP25. D, CDC42. And E is ROA, which is known to have an effect on neurite outgrowth due to overexpression. So this technique of high-content microscopy ties into the Module 4 protein expression and analysis by providing a powerful tool for studying cellular processes, protein-protein interactions, and subcellular localization. It allows researchers to investigate the effects of genetic manipulations on cellular phenotypes and provides quantitative data for studying signal pathways, cellular responses, and disease mechanisms. So, limitations of high-content microscopy are the potential for phototoxicity and photobleaching. So phototoxicity is damage to cellular macromolecules upon excitation, which can impair sample physiology and lead to sample death. Photobleaching is the process upon illumination with high laser power over prolonged periods of time. The imaged organisms are damaged, and the decay of fluorescence intensity caused by light excitation. So basically, it is due to the prolonged exposure of cells to excitation light during image acquisition, which can affect cell viability and result in inaccurate data. Another limitation is the need for careful optimization of imaging conditions and analysis to minimize variability and ensure reproducible results. This is also a large amount of data that is generated by high-content microscopy, and it requires sophisticated tools and computational resources. Image 2, this is showing scanning electron micrographs of adult Drosophila eyes compared to the wild type, which is on the left. The transgene led to a rough eye phenotype with misarranged and fused ommatidia. And that is in image A. In image B, it is high-resolution images of BSC1 cells stained for DNA and alpha-tubulin, which shows the effect of monostrol treatment on spindle formation. In image 3, it is HCT116 cells stained for DNA, alpha-tubulin, and actin to extract quantitative phenotypic features describing cell morphology. In D, it is Drosophila cells stained. As in image C, with RNAi-mediated knockdown of Rho1 leads, which led to enlarged multinucleated cells. In image E, there's confocal micrographs of A549 cells infected with influenza virus and stand for the virus and the CD63. where the yellow indicates the virus particles co-localizing with CD63 labeled latent zones. And in image F is the confocal micrographs of L cells after treatment with WNT3A conditioned medium. The nuclei are stained, and formation of lipid droplets are visualized with BODIPY 493503 staining. So what are the advantages of high content microscopy? It includes its ability to analyze multiple parameters simultaneously, providing a comprehensive understanding of cellular events. So compared to other techniques like traditional microscopy, high content microscopy offers high throughput capabilities and enables the analysis of large sample sizes and screening of compound libraries. It also allows for quantitative assessment of cellular phenotypes and offers a more objective and standardized approach compared to manual scoring or qualitative observation. For my image 3, this is showing the performance of deep block for classifying images of cells expressing ORF-RFP fusion proteins collected for manual assessment. So in image A, it is showing the micrographs from a screen of wild-type B cells expressing ORF-RFP fusion proteins. That localize to 10 unique output classes. In B, it is showing the transfer learning, where all layers except for the last layer in red are initialized to the network trained on the ChunLab dataset. In C and D, it is showing comparison of classification accuracy, which is the y-axis, and different training set sizes, which is the x-axis, when transfer learning is implemented using deep block versus training a network from scratch, which is the blue line, and deep block is the red line. And the error bars indicate the standard deviation of accuracy. It also shows the confusion matrix for transfer learning, the deep block network trained on the dataset to the new dataset with 100 samples per class. And that's mostly in image D. And this is using 100 samples per class. Controls that are used for high content microscopy include negative control, which involves imaging cells that are not treated with any fluorescent probes or stains. And it's basically a baseline to assess background fluorescence. Positive control, which is it's treated with a known compounder subjected to a specific treatment that induces specific cellular response or phenotypes. And it helps validate the functionality of the system. And an internal control is also used, and this is meant to be used for reference dyes or beads to ensure consistent imaging settings and quality across different samples. And it helps to monitor and correct variations in imaging conditions. Here are my citations. Thank you.